Right. So hello everyone. I am Apurva Chitre and I'm from the Pharma Lab at the University of California, San Diego. And today I will be talking about the genetic analysis of multiple measures of locomotor activity. And so the rats that we've used for this study are part of a large multi-site project which focused on genetic analysis of behavior phenotypes which are related to drug abuse. And so the heterogeneous stock rats were sent to three different phenotyping centers throughout the US for phenotyping, and the rats were exposed to a different battery of behavior testing at each of these sites. And the Pharma Lab was involved in genotyping and performing genetic analysis of this behavior traits. So then, as part of this big project, locomotor traits were also measured in these phenotyping centers in different rats. And so I wanted to point here that the Tennessee cohort included adolescent rats. And so these rats were much younger than the Michigan and the New York cohorts. And so the combined sample size for these three phenotyping centers is approximately 4,200 male and female heterogeneous stock rats. And I also wanted to point out that these cohorts differed in procedural details, like the size of arena. For the Tennessee Center, the rats were placed in a much larger open field chamber as compared to the Michigan and New York cohorts. And the Michigan and the Tennessee cohorts also used an automated video analysis software, whereas the New York cohort used beam breaks to track the behavior of the rats. And the duration of the test is also much longer for the Tennessee cohort as compared to the Michigan and New York. So there were procedural differences between the different cohorts. So we genotyped greater than 6 million SNPs per individual using the genotyping by sequencing method. And we estimated the genetic correlations, SNP heritability estimates using GCTA. And we also used GCTA for mapping. And we used permutation-derived significance threshold. So one of the questions that we've addressed is, are the locomotor traits heritable? And the answer is yes. These do exhibit high SNP heritability, and these range from 0.2 to 0.3. Another question that we can ask is the genetic underpinning of these locomotor traits measured in different phenotyping centers, is it similar or is it different? And so what we've estimated is that the Tennessee Center showed the lowest genetic correlations with Michigan and New York, whereas the genetic correlation between the Michigan and New York locomotor traits was pretty high. It was 0.6. And so we think that one possible explanation for this low genetic correlation for the Tennessee locomotor trait could be that because of the open field locomotor activity in Tennessee includes a measure of anxiety, while the locomotor activity measured in a small box for the Michigan and New York cohorts, it does not include this component. Another explanation is that in Tennessee, the animals are adolescent, and we think that maybe during this developmental stage, the mechanisms underlying locomotor activity could be different. So we've also mapped these three locomotor traits. And so this is a combined Manhattan plot for the GWAS data for these three locomotor traits. And you can see that we've identified a total of 6 independent QTLs. And so these are colored by the top SNP for each of the QTLs for the three traits. Now let's zoom in into one of the QTLs. And so this one is on chromosome 2 for the Michigan locomotor trait. And so now we have zoomed in to just a few megabase on chromosome 2. And on y-axis, we can see the significance of the association. And we can also see the genes which are present in this QTL interval. And the focal dot is the top SNP for this QTL, the one with the most significant association. And the color of the dots represent the LDR square of the top SNP with the other neighboring SNPs. And so the red color indicates that these SNPs are in high LD with the top SNP. And so in order to determine the QTL intervals, what we've done is we have identified those markers which are in high LD with the top SNP. And so we've used a cutoff of R squared greater than or equal to 0.6. And so on the basis of this criteria, the interval size that we've determined for this QTL is 1 megabase. And in this 1 megabase interval, there are two genes. One of them is an uncharacterized gene, LOC gene, and the other one is this catenin delta 2 gene. And so the catenin delta 2 gene provides instructions for making the delta catenin protein. And this protein is active in the nervous system, where it helps in cell adhesion and it plays a role in cell movement. And in humans, this particular gene has been associated with autism and ADHD. So we think that this could be a potential candidate gene, which could be driving this association at this particular interval. All right. So before we talk about the next QTL, I wanted to talk about this RNA-seq data that we have, which was measured in 5 brain regions. And Daniel Munro from our group, who also gave a talk yesterday about this data set, has worked on calling eQTLs or expression QTLs for this particular data set. So now let's zoom into this chromosome 18 QTL for the New York locomotor trait. And so there are 10 genes in this interval. And the question we can ask is, are there any eQTLs that co-localize with the behavior QTL? And so for this same chromosome 18 locus, we can see that there are significant eQTLs, and it is associated with regulation of these genes in brain tissues that might be relevant for this locomotor trait. And so we think that this could be one of the things that might cause this locus to be associated with the locomotor behavior. And so if you notice, we've also calculated the LDR square of the eQTL top SNP with the GR top SNP. As you can see, for this particular gene, the R square is pretty high, it's 0.9. So we think that this could be one of the genes that may be causally related. So another question we can ask is, what other traits also show association at this same locus? And so this analysis is part of the PHEVAS pheno-wide association analysis. And so what we've done is we've looked at traits that also co-map in this 3 megabase window. And so we can see that there are some traits that co-map to the same region. And the peak SNP for these traits are in high LD with each other. Okay, so it's plausible to think that they are causally related to one another. And so the traits that I have highlighted here are from the same behavior paradigm, locomotor testing, and they are different measures of locomotor, which are activity and rearing. But the interesting one that comes up is the nicotine self-administration trait. And so from this, it shows that the locomotor behavior is associated with a more complex drug self-administration trait. And so this might make us think if there is a relationship between locomotor behavior and drug self-administration. And this kind of association might help us to interpret the meaning of the traits that we are looking at. And it will also help us to design follow-up studies about it. So we think that there is something about this particular locus that is influencing complex behavior traits. So in conclusion, we identified a total of 6 independent QTLs for 3 different locomotor-related traits. And we observed strong genetic correlations for locomotor traits between the Michigan and New York cohorts, but not for Tennessee. And the Tennessee Center, which showed the lowest correlation, it included younger animals, it used a larger open field, and it also included a longer test than Michigan and New York. And so now we are also reprocessing the data from the Tennessee cohort to focus on a shorter time period. And we are also analyzing a much larger data set. And so we think that with increased power, we will see if some of these loci are sex-specific. So I'd like to thank all the PIs and everyone involved in this work from the P50 NIDA Center. And thank you so much for listening.